Dear participants, very welcome. On behalf of me, I'm the chair of the EFLM working group on test evaluation, and it's an honor and a privilege to introduce you to the Unmet Clinical Needs Assessment webinar. We have to do this for proper biomarker evaluation, and Dr. Monaghan will explain us the practical toolbox which has been developed for our lab profession in order to do this in a proper way. But before starting, I would really like to thank Danielle Ryder for arranging and managing all the practical things to make this webinar happening. And of course, our guest speaker, Dr. Phil Monaghan, who is going to take us through this webinar. But first, a few introductory words. Dr. Phillip Monaghan is a consultant clinical scientist at the Christa Pathology Partnership. After graduating from the University of Manchester Institute of Science and Technology in biochemistry with applied molecular biology, Phillip went on to complete his PhD in enzymology at the University of Leicester. He subsequently commenced training in clinical biochemistry at Wirral University Teaching Hospital, Alder Hey Hospital, and the University Hospital of South Manchester. He has been a clinical scientist at the Christa Hospital and more recently at the Christa Pathology Partnership in Manchester, UK for the past 9 years, obtaining fellowship at the Royal College of Pathologists in 2014. Phillip is a state-registered clinical scientist with the Health and Care Professions Council and a European specialist in laboratory medicine. He is also a honorary lecturer at the University of Manchester in the Faculty of Medical and Human Sciences, at the Institute of Inflammation and Repair, and he regularly teaches on the Master's Clinical Science program. He is a member of the UK NAQUA Steering Committee for Immunochemistry, and he is also an associate editor for the Annals of Clinical Biochemistry. He is a consultant member of the European Federation of Clinical Chemistry and Lab Medicine Working Group, to which I referred already, for test evaluation. His special interests include evidence-based medicine, among them test evaluation, also biochemical endocrinology and neuroendocrinology, and assay interference studies. He was awarded the Silver Research Medal from the Royal College of Pathologists in 2012 for his work on serum cortisol measurement in Cushing's disease. He is a lead healthcare scientist for his hospital. So, Phil, the floor is yours, and we are looking forward to this webinar very much.
Thank you, Christa, for that kind introduction. And good evening, everyone. Thank you for attending this latest EFLM webinar. I have the pleasure to discuss unmet clinical needs assessment for biomarker evaluation at practical toolbox for laboratory medicine. I'm based at the Christa Hospital in Manchester. It's the largest single-site cancer centre in Europe, treating over 44,000 patients per year. Pictures of the Christa, and also at the bottom right is the Manchester Cancer Research Centre, the Oglesby Centre, just across the road. So we have a very close-knit campus, very close collaboration with the University of Manchester. The University of Manchester is also the main hub for clinical scientist training in the UK. And just before I go on to my presentation, I had the opportunity to join the EFLM working group for test evaluation back in 2012 as a young scientist member. It's really given me the opportunity to work with excellent scientists across Europe and across the globe, really, enabled me to travel and present research. So to any early career scientists joining us, I really urge you to get involved with your national societies, get involved with the EFLM, and grasp any opportunities that come your way, because they can be really rewarding. So I'll start the presentation today just to discuss a brief introduction to the working group for test evaluation and some of our ongoing work in this area. The challenges and opportunities of test evaluation and the evolving landscape, and put that into the context of the new in vitro diagnostic regulations, which we're currently in transition, and then move on to the practical toolboxes that the working group for test evaluation is developing and published, both in terms of assessment and performance specifications, both analytical and clinical performance specifications. So the working group itself is a collaboration jointly between the EFLM and also the Australasian Association for Clinical Biochemistry, the AACB. It's a collaboration between laboratory professionals, clinical epidemiologists, health technology assessment experts, more lately health economics, and also industrial IPD partners as well. So it really is a truly multi-stakeholder collaborative working group. We aim to improve health outcomes and healthcare efficiency by better medical tests and by developing guidance and practical tools for biomarker development and test evaluation. We should all be familiar with this brain-to-brain loop. Eric Kilpatrick and Sverre Sandberg recently published an overview of EFLM working group activities across this brain-to-brain loop. And I've just placed this slide on here to illustrate that the test evaluation working group really focuses on the analytical aspect of this brain-to-brain. As the working group, we've also recently provided 2 very successful test evaluation courses over the last couple of years. The first one was in Leiden in the Netherlands towards the end of 2016, developing medical tests to improve patient outcomes. The second, more recently, was in Sydney, Australia last year, assessing the value of biomarkers as an interactive workshop. Our activities can also be found through the EFLM webpage for the working group test evaluation. Here you can find our published articles with links to those, and also links to our practical toolboxes, including our interactive checklist that I will move on to later on in the presentation. So just to place this in the context of biomarker research and development and where test evaluation really can play its key role, I did a search recently of PubMed using the search term biomarker, and I found over 800,000 hits. And if you run this by year of publication, you can see that it really is increasing year on year. So biomarker research and development and biomarker discovery really is a truly growing market. Yet a publication by Drucker and Krapfenbauer and EPMA showed that though there is an increasing number of presentations, publications in this area of biomarker discovery, the number of biomarker patents submitted, and also market approvals remains pretty stagnant. So there seems to be a translational inertia from biomarker discovery through to translation and clinical utility. And this was really illustrated quite eloquently by Lee Anderson in a paper in Clinical Chemistry back in 2010, where he did a review of FDA-cleared protein diagnostics. And he found that the FDA-cleared protein biomarkers at a rate of 1.5 per year. And this really was judged to be insufficient to meet developing clinical needs and demands. So we really need to improve biomarker discovery and reduce research waste in this area quite significantly. And George Post, writing in the journal Nature in 2011, also described this really as a drop in the ocean. There are hundreds of thousands of newly discovered biomarkers in the scientific literature, yet a very small proportion actually make their way translationally through to the clinic for clinical use. And again, in clinical chemistry, in a very useful Q&A, the expert panel alluded to the fact that despite a 15-year effort, there's been no major disease biomarkers discovered using mass spectrometry-based proteomics. Quite tellingly, the main obstacles are well-designed studies that ask the most important clinical questions. So you can see there are major challenges and opportunities to be had here in terms of biomarker discovery and test evaluation. So moving on to these aspects, some major challenges include the introduction of new tests with poor evidence base, which has been all too prevalent in the past, variation in the adoption of tests across conurbations, under and over utilization of tests. You can see that through the publication of the Atlas of Diagnostics dermatologists, for example. There's much variation across the UK. Slow adoption of new tests, so you can get early adopters, but this isn't necessarily translated across different clinical arenas. And also, ultimately, tests need to be employed correctly across clinical pathways, and they need to fit local needs, local clinical needs. So, tests really, in an ideal world, provide information to provide clinicians to make better decisions about the care of their patients, and that then, further down the line, impacts on patient management and ultimately patient outcomes. But the evidence base for testing is relatively poor in this area because the interventions are usually not directly linked to the test themselves. So you need to link the evidence. So we've started to look at this area. The test evaluation group have published key definitions and also a cyclical test evaluation framework to really start with a good foundation, if you like. And we published our first special report back in 2014 in Clinical Biomarker from biomarkers to medical tests, the changing landscape of test evaluation. And within this really key paper, we devised a cyclical framework for test evaluation, viewing both analytical performance, clinical effectiveness, cost effectiveness, broader impact. And this is an iterative and dynamic evaluation cycle, really driven at the heart by the clinical pathway. So the clinical pathway and the test purpose and the test role within that clinical pathway is very important to guide analytical, clinical performance, clinical effectiveness. Now, within this paper, we provided key definitions for IVD test evaluation. And as an example, we provide the proposed term, so analytical performance, for example. We also provide related terms which you might find within the literature. So we take analytical performance as an example. You may also see this as described as analytical validity or technical efficacy. We also provide an explanation or a definition. So for analytical performance, this is the ability of an in vitro medical assay to conform to predefined quality specifications. Those specifications, for example, being the Milan mandate from the 3-model approach from the 2014 EFLM strategic conference, looking at analytical performance specifications based on clinical outcomes, based on biological variation, or based on state of the art. And we also provide illustrations within this scoping paper as well. So for analytical performance, again as an example, the universal definition of myocardial infarction recommends that high-sensitive troponin assays must have accurate in precision to within or equal to 10% coefficient of variation of 99%. We also include the key definitions of clinical performance. That is the ability of a biomarker to conform to predefined clinical specifications in detecting patients with a particular clinical condition or in a physiological state. Clinical effectiveness within the dynamic test evaluation framework is the ability of a test to improve health outcomes that are relevant to the individual patients. Cost effectiveness within the cycle is the analysis which compares the changes in costs and in the health effects of introducing a test to assess the extent to which the test can be regarded as providing value for money, and broader impact, which needs to be included within the test evaluation framework. This is the consequences of testing beyond clinical effectiveness, beyond outcomes. So are there any additional points to consider in addition to clinical effectiveness and cost effectiveness? These may be acceptability of the test, social aspects, psychological aspects for the patient, legal aspects in terms of, say, next generation sequencing, etc., ethical aspects likewise, societal aspects, and organizational consequences. And anything else you can really think of considering within that test evaluation framework. So moving on to IVD medical tests within the context of the changing regulatory environment, it's first off just important to acknowledge that IVD medical tests differ from other medical devices, and that the risk and benefits they pose are related to the impact on patient management rather than direct contact between the device and the patient itself. So it's tests driving downstream clinical management decisions to hopefully improve patient outcomes. So the risk there is indirect, but there is a risk. And the new IVD regulation came into force in April 2017 into legislation, and we're currently undergoing a transition period from the previous legislation. So you'll be familiar with the In Vitro Diagnostics Directive 9879 EC, and the transition to the future legislation, the Medical Device Regulation, and more importantly the IVD Regulation 2017-746, is a 5-year transition period. So the new IVDR will actually come fully into force in May 2022. Within the IVDR, they stipulate what is known as a performance evaluation plan, and this takes into account the EFLM working group's diagnostic test evaluation framework, if you like, including aspects of analytical performance and clinical performance. And the performance evaluation plan for different tests also includes, well, it specifies the characteristics of the test, of the IVD, but importantly includes and considers that these evaluations are continued during the life cycle of the in vitro diagnostics. This is post-market surveillance, if you like, or post-market performance follow-up. And this is the planned monitoring of scientific developments and changes in medical practice by the manufacturer during the life cycle of the IVD, if you like. And this is really important. If we take examples, the top molecule on the right here is, you may recognize it, it's estradiol. The lower molecule is a similar steroid-like ring structure, but this is fulvestrant. Now, fulvestrant is a selective estrogen receptor degrader, so it's an estrogen analog, basically, has a long aliphatic hydrophobic side chain with a sulfonyl fluoro moiety, so it makes it really hydrophobic as a selective estrogen receptor degrader. And this is indicated for treatment of breast cancer patients which have previous endocrine therapy, metastatic disease in postmenopausal populations. Now, we published a paper very recently in the British Journal of Cancer in February of this year, and we found that in breast cancer patients taking fulvestrant, there was significant cross-reactivity in immunoassay platforms. So the middle lane here is AA, that's the Abbott Architect. SC to the right denotes the Siemens Sensor platform. So these two immunoassays performed quite poorly when compared to the gold standard LCM-SMS, tandem mass spectrometry. And this is important because for clinical trial eligibility for breast cancer patients, a comprehensive assessment of menopausal status, including biochemical assessment, which it also includes serum estradiol measurement, is absolute paramount for entrance into clinical trials. If patients are taking fulvestrant, this can confound the biochemical assessment of menopausal status, giving spuriously raised estradiol levels, and this can have a huge impact on patient outcomes potentially. So we really published this brief communication to ensure that oncologists in this area of patient care are fully aware of the implications that fulvestrant can have potentially on estradiol measurements. Likewise, we also published a paper in the Annals back in 2011 looking at cortisol measurements, specifically in patients with Cushing's disease, medically managed by metarapone treatment, which is an 11-beta hydroxylase inhibitor. Now, metarapone has the effect of shutting down the adrenal glands in terms of cortisol production, but that has the consequence of increasing cortisol precursor in circulation, such as 11-deoxycortisol. And we found that 11-deoxycortisol cross-reacts with certain cortisol immunoassays. You can get spuriously raised cortisol when patient is on metarapone therapy. And this can have adverse knock-on consequences for the patient because cortisol measurement is important throughout the day in order to titrate metarapone dose. So if you're reporting a spuriously raised cortisol from an immunoassay, this can potentially lead to over-titration of metarapone, which at worst can be fatal. So to mitigate potential for erroneous clinical decision-making concerning dose titration for meteropon therapy, we advocated that cortisol should be measured by tandem mass spectrometry for patients specifically receiving meteropon therapy, and we've implemented a pathway to that end at the Christa Hospital. Now, going back to the performance evaluation plan, this is also a specification, if you like, for the intended purpose of the device, and this links in quite nicely as well with our scoping paper in the fact that we provide key definitions for IVDs in terms of test purpose, and that is a description of the intended clinical application for the test and how the test information can be used to improve clinical management in practice. Examples of test purpose are a diagnostic test, a prognostic test, and a screening test, for example. And we can say that HbA1c is a diagnostic test for diabetes. Now, the test role, again, is all important within the clinical pathway, and that describes how the test used within this for a specific clinical purpose or intended use will be positioned within that existing clinical care pathway for a specific condition within a target population. So where exactly within that pathway is the test going to reside? It could be a replacement test, so replacing CKMB being replaced with troponin, a more contemporary biomarker for ACS diagnosis. It could be a triage test, and I've given the example here as brain natriuretic peptide, BMT, as a triage test prior to echocardiography in the congestive heart failure pathway, or it could be an add-on test. Perhaps if you do a thyroid function test or a TSH and a free T4, which is abnormal, you may want to add on a free T3 if you're thyrotoxic. Going back to our test evaluation framework within our scoping paper, it can also be used as an audit tool, and this was exemplified in a 2017 paper in BMJ Open, published from the Oxford Diagnostic Horizon Scanning Program. And they mapped 500 primary research papers in point-of-care test evaluation to our test evaluation framework. And they found that less than 20% of papers reported on clinical effectiveness, comparative clinical effectiveness, cost effectiveness, and broader impact. So there's quite a paucity in the scientific literature within these key aspects of test evaluation. And they also found that a test evaluation cycle, on average, takes about 9 years, to complete. So it's quite a long, protracted process. Again, the test evaluation cycle really is driven by the clinical pathway, but it's focusing on the unmet clinical need within that pathway. And to enable unmet clinical need assessment to be done correctly, we need to really look at mapping the pathway to visualize it. So it helps to visualize the complexity of clinical pathways for different stakeholders. If we map the flow of the clinical pathway, it helps us to assess the extent of the clinical problem. And it also, rather importantly, helps us to visualize the current pathway and quantify any impact of innovation, either a new biomarker or doing things slightly differently. Quality improvement as part of our development processes within the laboratory, and helps exemplify the importance of laboratory medicine to our colleagues in our clinical care setting. And a really good example of this is from a paper published in Circulation just last year. This paper looked at a study of cross-system implementation of a national clinical care pathway across a number of hospitals for acute coronary syndrome. And this paper reported on 7 acute care hospitals in New Zealand, looking at over 31,000 patients investigated for suspected ACS with serial troponin measurements. They implemented a clinical care pathway, with a documented pathway, serial troponin testing within 3 hours of arrival of the patient, and directions for combining risk stratification and ECG and troponin testing in an accelerated diagnostic protocol. So this new pathway was monitored for over 4 months and then compared with standard of care for the 6 months preceding that implementation process. And in terms of outcomes, they found that for a primary outcome, the mean 6-hour discharge rate increased from 8.3 to 18.4%, so an improvement in the discharge rate. The odds of being discharged within 6 hours increased after the clinical pathway implementation, with an odds ratio of 2.4%. In terms of secondary outcomes, they also found that patients without acute coronary syndrome, the median length of hospital stay decreased by 2.9 hours on average. So some very beneficial outcomes for patients, both in terms of primary and secondary outcomes. Importantly, the paper also noted that patients discharged within 6 hours, there was no change in 30-day major adverse cardiac events. So there was no additional risk implemented there. So clinical pathways do really afford us an opportunity for sharing best practice, for diagnostic stewardship, and also an opportunity for standardization across pathways. And that really also is relevant across networks as well, which is a growing area, if you like, within laboratory medicine. Joined up networks of laboratories across geographic conurbations. So really it is an opportunity for sharing best practice and standardization across the pathway. They can also be a catalyst for biomarker development. Pathways can convince policymakers and commissioners that newly developed tests are worthy of funding. They can guide desired analytical and clinical test performance specifications, as I'll move on to a little later in the presentation, and also guide medical decisions that will lead to improved patient outcomes. And that can be illustrated within the clinical pathway prior to implementation. If there's really strong links between clinical management decision, the test. So moving on to the practical toolbox for laboratory medicine that the EFLM working group for test evaluation has to offer. Firstly, I'll just move on to the unmet clinical needs assessment. So as I've mentioned, we have the cyclical test evaluation framework. The purpose and role of the test must be defined to address an unmet clinical need within that clinical pathway. The group has defined that an unmet clinical need is any missing or inadequately performing component of the clinical pathway. So we really need to be asking, what is the clinical management problem at the outset? And we've published a number of articles in this area recently. The first paper from the working group was back in 2016, published in CCA, Biomarker Development Targeting Unmet Clinical Needs, where we first presented the checklist of unmet clinical needs assessment, and this was followed shortly after by a follow-up paper in the EJIFCC, a practical guide to identifying unmet clinical needs for biomarkers. And our unmet clinical needs checklist is really a very simple 4-step checklist. At step 1, we want to identify the unmet clinical need for a test, verify the unmet clinical need for the test in step 2, and in step 3, validate the intended use or the purpose of the test, and finally in step 4, we need to assess the feasibility of using the test in clinical practice. And we elaborate on these 4 steps by asking 14 key clinical questions. So in step 1, we start by asking, what is the clinical management problem and desired outcomes? So what are the health conditions and clinical management problems? What is a test group? What is the, so we're taking into consideration the target population there. What is current practice? What are the limitations of this current practice? And what are the desired outcomes that we're wanting to achieve? In order to verify the unmet need, we need to be asking, are there any existing solutions within the existing clinical pathway? Could the problem be solved by optimizing current practice within the pathway? Could these solutions, possible solutions, be effective if there are any? Could they be cost effective, and are there any barriers for these solutions? We then want to validate the intended use of the test. So would the biomarker contribute to the solution? How could the biomarker alter and improve current practice? What are the expected outcomes from the test results? And how do these outcomes compare to the desired outcomes defined in step 1? Both in step 1 and step 3, we need to be looking at mapping the pathways, both in terms of current clinical practice and projected future clinical practice with a new biomarker or new testing strategy. And then finally, we need to look at feasibility of the and implementing biomarker within clinical practice. So there's a biomarker solution that has been identified and proposed feasible for clinical practice. Under what conditions would the new biomarker be feasible? Be that commercially feasible, economically feasible, technically feasible? Have we got enough of the technical competence, staff resource, staff expertise to implement it, and also organisationally? And are there any other barriers, as alluded to perhaps in our test evaluation framework? So it complements the test evaluation framework rather nicely. And with the help of Daniel Rajdal in his capacity as the chair of the EFLM distance learning and e-learning group, Daniel has been absolutely central to helping us to translate our unmet clinical needs checklist into an interactive format, which is now available on the e-learning platform. Also, quite helpfully, Daniel has put together a short YouTube video to show how to use the unmet clinical needs interactive checklist to make things slightly easier. Also, within this interactive checklist, we've incorporated the key definitions within the original scoping paper. And here I've given the example of biomarker. If you click on biomarker in the interactive checklist, a pop-up occurs whereby it gives you the definition of the biomarker and also provides examples. And these key terms, unmet clinical needs, clinical performance, analytical performance, biomarker, IVD, etc., these are peppered throughout the interactive checklist, and you just click on those. It gives you the definition and some really good key examples, so everyone's nice and clear on a consensus terminology. The interactive checklist provides a tutorial for how to go about using the checklist. We also provide pathway mapping templates so that you and your stakeholders can map out current pathways, new pathways, and we provide model answers as well to give illustrative examples on how to use these pathway maps. The example given in the interactive checklist is focused on high-sensitive troponin within an acute coronary syndrome pathway. And we also provide an unmet clinical needs checklist, so you can fill in the details as you go through steps 1 to 4 within the checklist. And we also provide an example within the checklist as well to discuss with your colleagues. And these examples just given here on the left-hand side, we've got pathway map number 2, where it shows target population and the clinical setting, current test, the new biomarker to replace the current test, the management decisions, both for the existing and new pathway, health outcomes and unmet clinical need, and also health outcomes for the new test in terms of potential benefits and potential harms. So the pathway mapping exercise is very comprehensive and asks a lot of key clinical questions to help robust adoption of new biomarkers. And also on the right-hand side here, I've just given step 1 of the interactive checklist. So this asks the checklist asks those specific key questions that I've just been through, but in an interactive format, so you can fill out the online version as you go. And also there is a feedback form and discussion forum, so you can have the opportunity to submit your completed clinical needs assessment interactive checklist completed forms for review by the EFLM working group. We also would encourage you to fill out the feedback form in terms of how user-friendly you found it, what were your initial reactions, and this will help the working group to update and iterate this unmet clinical needs assessment for future use to improve its utility, if you like. Moving on to performance specifications, this is an area that the working group for test evaluation has also undertaken considerable work. Firstly, analytical performance specifications, or APS. These were the topic of the first EFLM strategic conference held in Milan in October 2014, 15 years after the Stockholm conference. And the Milan mandate really simplified the aspects of APS and stipulated a 3-model approach, whereby model 1 is looking at APS based on clinical outcomes, be they direct, as in model 1A, or indirect, model 1B. Model number 2 is based on biological variation of the measurand, and analytical performance specifications based on model 3 is state of the art. And really critically and quite helpfully in the consensus statement from that EFLM strategic conference, the consensus statement stated that the approach acknowledges that the intended use, so the purpose of the test, may modulate the selection of the best approach for analytical performance specifications. So they acknowledge that there is an intrinsic association between the test purpose and which approach you take to defining analytical performance specifications, which is incredibly helpful. The working group for test evaluation also provided an opinion paper for this 2014 conference based on Model 1 outcome studies, asking the question, is it possible? And we also allude to how you can go about defining APS based on outcome studies, be it through the linked evidence approach, be it through simulation models, decision analytics. And as an example here, you can define performance criteria for APS based on Model 1B, which is an indirect approach, because Model 1A is very difficult. It often requires randomized control trials to get direct links between test and patient outcome. So here, Staub, publishing in BMC Medical Research Methodology back in 2012, really illustrated the linked evidence approach here, whereby if you have diagnostic accuracy of the test to classify patients for the clinical decisions, you can then link that to clinical decisions if they are well defined, and link that to consequences of classification if they are well established. So you can link different studies to enable analytical performance specifications based on clinical outcome assessment. Similarly, you can use simulation modeling, and I'll just give this example from clinical chemistry back in 2014. This paper quite eloquently looked at the effects of measurement frequency on analytical quality required for glucose measurements in the ICU by simulation modeling. And they found that adverse effects of imprecision markedly blunted when glucose was measured every 5 minutes rather than every hourly. So if you compared a regimen whereby you had a glucometer with a 5% CV when measured hourly, this performed quite similarly in terms of the frequency of hypoglycemia when measuring a glucometer with a 10% CV with measurements taken every 5 minutes. So varying the analytical performance specification can have different consequences on patient outcome. Now moving on to clinical performance specifications. We've done a lot of work in this area as well, and quite thankfully, hot off the press, I can report that last week, the working group test evaluation published in the analytical in the Annals of Clinical Biochemistry. This paper, just last week, so it's published ahead of print, based on setting clinical performance specifications to develop and evaluate biomarkers for clinical use. And in this paper, the working group provides a practical guide for setting minimum clinical performance specifications to strengthen clinical performance study design and interpretation. And we also provide a 5-step approach to using test evaluation and decision analytic frameworks. And we define CPS to set criteria that quantify the clinical performance of a new test to attain and allow better health outcomes than current practice. And we also classify the proposed benefits into 3 broad groups and describe how to set minimum CPS at a level where the potential harm of any false positive or false negative results does not outweigh the benefits to the patient. And we illustrate this with key examples in the paper. This table is taken from said paper, and we show the 5-step approach, defining intended benefit at step 1. So what patient or other hospital or other benefits do you hope to achieve by using this biomarker? In step 2, we map the current pathway. So you can see this complements very well the unmet clinical needs assessment checklist. So what patient group will be tested? What tests do they currently receive to detect and target condition? What key actions are based on the results? Any further tests or treatments? And what are the benefits and the harms of these actions? So it's really mapping out again the clinical pathway so that stakeholders can see the flow chart from population to outcome. We also include step 3, proposed new test role. So as I said, we divide this into 3 broad groups: add-on test, triage testing or replacement testing. Step 4, linking clinical performance requirements to intended benefits. And finally, in step 5, setting minimum acceptable clinical performance levels. So if you take the replacement test, for example, no trade-offs, benchmark against existing test accuracy. And once you have these requirements, you can then use these minimum clinical performance levels in a future test accuracy study to assess whether they are acceptable or not, whether to accept or reject for further work. Likewise, in the paper, we provided 4 examples using different test roles. If we look at the FIT test, the fecal immunochemical test, for example, this is really a test to improve more accurate and early detection of colorectal cancer, replacing the existing FOBT. In terms of step 4, clinical performance requirements, we need to be looking at more true positive results with an acceptable false positive rate. So potential harm there may be more false positives leading to higher colonoscopy rates. And the approach really in this context for a replacement test is benchmarking the existing test accuracy of the FOB test in terms of diagnostic sensitivity and specificity in a representative patient population of intended use. So to summarise really, the performance specifications should reflect clinical needs of patients within the clinical pathway, tailored to the purpose and role of that test within the clinical pathway, and ideally we need to have a standardised clinical management decisions to maintain a strong, valid and reproducible link between test performance and outcomes. And that is also really important when you're using the linked evidence approach in Model 1B of the Monaghan hierarchy of APS as well. So some key take-home messages here. We need to identify how novel biomarker-based testing strategies can change existing clinical pathways, ideally by mapping current clinical pathways to identify areas of unmet clinical need and utilising that unmet clinical need checklist. The corollary of this approach, which I've discussed, is the definition and specification of analytical and clinical performance criteria to guide the evaluation studies and the cyclical manage based on the cyclical test evaluation framework, keeping the clinical pathway and the test purpose and test role and ultimately patient outcomes as the key drivers in the whole process. And there's really a major opportunity here for laboratory professionals to really play a key role in the development and implementation of clinical pathways for new and existing laboratory tests within your hospitals. And that's collaborating with key stakeholders, clinicians, IVD, research and development, patient advocates. So now we invite researchers and laboratory professionals to use these guides and use these toolboxes, if you like, to help establish clinical need, set minimum levels of performance to strengthen study design, interpretation, and reporting research groups. The approaches describe really with key biomarker stakeholders in terms of R&D discovery researchers, clinicians, lab professionals, IVD industry, as well as the patients and healthcare policymakers. And we hope that the practical toolbox and the online checklist, for example, can help facilitate these interprofessional discussions and lead to more efficient biomarker translation into clinical practice. I thank you for listening, and I'm happy to take further questions. So back over to you, Christa. Thank you very much.
I repeat, thank you very much, Phil, for this webinar. Can you understand me?
Thank you, Christa. Yes, I can hear you now.
Okay, so it's open for discussion, and I asked the public whether there were, or the participants, whether there were any questions on this clinical needs assessment. If not, I wanted to ask you a specific question, Phil. We just saw this very important work, and you showed very nicely the gap which we experience between biomarker discovery and implementation. Single biomarkers. What is your recommendation for the more complex omics type of biomarkers, which are also glorying at the scene and which seem very promising for precision diagnostics?
To get to grips with the test evaluation approach. But overall, it really is having a really good consideration of the clinical pathway and the test purpose and thoroughness test at the outset really. So as lab professionals, ideally we need to be working with biomarker discovery science, identifying unmet clinical needs at the outset to guide the analytical performance specifications of multiplex and omics markers in the trial setting. And as an example, I'm recently participating in a grant application for some new vascular response, increasingly prevalent both in terms of vascular response biomarkers and cytokine panels, for example, in my tertiary oncology setting, and the unmet clinical needs checklist very useful to understand where my collaborators are wanting to go with new biomarker development. So working closely with the clinicians, working closely with the biomarker development researchers, and to enable translation of work into clinical practice. So the unmet clinical needs can help to really help to design clinical trials for translational research.
Thank you very much. And what about lab-developed tests under the new IVD regulation? Yeah, if it develops a lab-developed test, then it's in fact its own manufacturer, so it also has to go framework with the same rigor that commercial suppliers have to go through.
Yeah, I mean, ideally, yes. I mean, it's quite a contentious topic at the moment. I know in the UK, the MHRA are looking into terms of exemptions for LDTs, assessment, analytical performance, clinical, etc. And really, for laboratory developed tests, it all starts scientific aspects underpinning the applications, clinical performance and clinical effectiveness, driving those key aspects of test evaluation in a cyclical and iterative process. So it really is a team approach.
Okay. No more questions from the participants? Then I think, Phil, we want to thank you for your major contribution, and Daniel also for the organization and management of this webinar. And maybe you have a last remark regarding the certification and the... This is for you, Phil. Yeah.
Yes.
Do you
some multiple choice questions. The EFLM e-learning platform. I participants to complete those questions and submit them through the e-learning.
all of you, thank you very much for attending this web. Especially thank to Phil, to the speakers. So, and we will take lessons from this marvelous message. Thank you very much.
Great. Thank you, everyone. Take care. Good night.